My name is Ben Finlay. I'm a research associate professor and director of tumor analysis at Sanford Burnham Prebys Medical Discovery Institute in La Jolla, California. I'm a cancer biologist. I did my PhD in Dublin, Ireland. I used to have an Irish accent. It's faded away now. I moved to La Jolla in 2003. I did my PhD in pharmacology. So we've always been interested in trying to find why drugs work for certain patients and not others. Cancer is obviously a disease of DNA, but people have mostly focused on genetic mutations. A lot of the samples we get are considered normal by karyotyping. There's technically nothing wrong with these patients, but of course there is because they have leukemia. So although their karyotypes and their cytobanding look normal, once we run this through the Sapphire system, we can detect structural variants easily and with just a level of granularity that's impossible with standard cytogenetic techniques. What we'd really like to do is track structural variants through the progression of the disease and associate this with drug response or drug relapse. Optical genome mapping can detect structural variants that are missed by much more labor-intensive and much more time-consuming methods such as karyotyping and cytogenetics, which require these very sort of arcane, difficult techniques and highly trained pathologists to do the analysis. With OGM, it's essentially a DNA isolation, a labeling, and then it's just loaded into the chip, run the machine, and watch it on your laptop. And with the Bionano Access software, you don't really actually need a bioinformatics specialist. You can literally grab the images out, stick them in a PowerPoint, and send them to your clinical partners. We've recently found that duplications of certain genes, particularly phosphatases, in certain leukemias, seem to be associated with resistance or sensitivity to clinically relevant drugs. So duplications of these phosphatase families seem to be associated with resistance to frontline drugs. So patients are going to get these drugs because that's what clinicians give them. Hopefully in the future, we will be able to stratify these patients to say, look, this group of patients here should get them, and this group of patients here, you just shouldn't bother because they're not going to respond. Just on a personal level, we want to sort of alleviate human suffering, and the more data we can get, the more informed decisions we can make. Our mission statement is, through science, the power to cure. So for us, finding out new things that people have missed before, things that explain why a person, in our research, why certain people respond to drugs, why certain people don't, and trying to ascribe these to the underlying genomic cause, I think that's what pushes us on to try and find as much as we can about these patients to eventually help.